The word synapse is taken from the Greek word which means a junction, and since we know that there is no anatomical connection or the continuity between the different neurons, they are functionally connected. Now, depending upon the different types of anatomical connections between the two neurons, they can be exodendritic, means that axon of one neuron will form a synapse with the dendrite of the another neuron. Then we have the exosomatic, axon of one synapsing with the soma or the body of the another neuron. And then we have this exoaxonic, which is the synapse between the axon of a neuron with the axon of another neuron. This is very less common type of synapse. The physiological classification of the synapse can be the electrical synapse, the chemical synapse, or even the conjoined synapse. First, talking about the electrical synapse, they are nothing but the transmission of a nerve impulse through these gap junction, and we know that these gap junction are connecting the two neurons, so that allows the easy transmission or the fast transmission of the nerve impulse. So this type of conduction is also known by the name of electrotonic conduction, and this type is seen in cases of one cardiac muscle to the another cardiac muscle. So they are basically connected by these clusters of ion channels, and therefore they are bidirectional in nature. This is seen in retina or even the olfactory bulb, and the connecting unit here is the connexon. So this type of electrical synapse offers high speed conduction to the neuron. Here you can see that same thing that we were talking about. They are connected with these connexons. They are in two states, the open and the closed. When they will be open, there will be flow of ions from high concentration gradient to the lower concentration gradient, resulting in the transmission of impulse. Now this is the chemical synapse, you can see, and it is the most common type of synapse which is seen in the central nervous system. So this, the neurotransmitter being the agent that carries the nerve impulse. So the information travels only in one direction, and they are fatigued. They can be fatigued. So this type of nerve synapse is present in type 1 and type 2 category. So we'll understand about this synapse. The conjoined synapse refers to a synapse where both chemical and the neural electrical type of transmission is seen. It is seen most commonly in the vestibular apparatus. It has speed as well as is the transmission by the neurotransmitters. Now coming back to the chemical synapse, which I said is the most commonest type of synapse. First, let us understand the structure of this chemical synapse. These are the different parts, that is synaptic knob, the presynaptic membrane, the synaptic cleft, and the postsynaptic membrane. So as I said, we'll be studying it under three headings: the presynaptic membrane, the synaptic cleft, and the postsynaptic membrane. And the synapse shown here is the exodendritic type of synapse. You can see here the axon terminal is terminating on the dendrite of the other neuron. Now coming first to the synaptic knob or the bouton. When we study through electron microscopy, we find that the presynaptic terminal, when they tend to approach the synapse, it loses its myelin sheath and divides into number of branches, which end up in small swellings which are known by the name of synaptic knobs. They are also known as end feet or also known as Boutons. So what is this? Basically, these synaptic knobs are nothing but the ending of an axon on the dendrite or soma or even the other axon. So they contain the neurotransmitter in the form of small and large vesicles. It contains mitochondria. It contains voltage gated calcium channels. So all these are present in the synaptic knob. So we can see here the small vesicles. The small vesicles are mainly containing acetylcholine or catecholamines, whereas the larger vesicles with a dense core contain the neuropeptide transmitters. So then we have the synaptic cleft. It is a small gap, maybe of 200 to 300 angstrom or even 20 to 40 nanometer wide. It is present between the presynaptic and the postsynaptic membrane, and it is filled by extracellular fluids containing glycoprotein. The neurotransmitter will be released into these spaces, either it is destroyed or taken up or used up in the postsynaptic membrane. Then we have this postsynaptic membrane. It is lining the postsynaptic process, and on inner aspect of the postsynaptic membrane there is a presence of zone of dense cytoplasm which contain the active zones of synapse. So it contains a number of receptors on which these neurotransmitters will be acting. The receptor proteins, we know that they can be the ion channels or they may be the enzyme type of receptor which are known by the name of metabotrophic receptors. So these are the receptors. The ligand gated channels on the postsynaptic membrane can be metabotrophic, that means they are linked to the G protein. So they can be G protein linked ion channels, cell enzymes, gene transcription, or even linked to the second messenger system. Or they can be ionotrophic, that means they are the plain ion channels which are linked to the influx or efflux of sodium ion, potassium ion, or even chloride ion. So when we talk about the transmission of the synapse, it follows three processes: release of neurotransmitter in the synaptic cleft, action of neurotransmitter on the postsynaptic membrane, and then termination of the synaptic transmission. If the nerve impulse is not terminated, it will go into the repeated circuit and that will lead to the fatigue of the neuron. So equally important is the termination of the synaptic impulse. So these are the different steps. First, whenever a nerve impulse reaches the presynaptic terminal, it will lead to the opening of voltage gated calcium channels, resulting in influx of the calcium ion. This influx of the calcium ion is responsible of activating the small G protein, which bring about the exocytosis of the vesicles. So, the movement of synaptic vesicles occur towards the presynaptic membrane, then triggering this cross-bridge cycle resulted in increased exocytosis. There is release of neurotransmitter, which is also known by the name of kiss and discharge type of neurotransmitter. The different type of snare protein, for example V snare protein or synaptic brevin, are responsible for this. After the release of neurotransmitter, it then diffuses into the synaptic cleft and the time lapse is less than 1 millisecond. So after that, again the membrane will be recycled back into the presynaptic membrane. In the synaptic cleft, it undergoes the three processes. Either it diffuses to the postsynaptic membrane to act on the receptors present there, or there is enzymatic degradation of the neurotransmitters, or it is actively transported back into the presynaptic membrane. The binding of neurotransmitter to the postsynaptic membrane or the receptor cause opening of channels through which ions can flow. So this can be excitatory or the inhibitory in nature, and therefore there is a development of EPSP, that is excitatory postsynaptic potential, and IPSP, which is inhibitory postsynaptic potential. Now let us understand this figure which has been taken from Guyton. Here in part A of the figure, you can see the resting neuron with unexcited presynaptic terminal is showing the resting membrane potential everywhere in the soma, that is minus 65 millivolts. Now come to part B. It shows that the presynaptic terminal has a secretory excitatory neurotransmitter. So when it is excitatory in nature, it will act on excitatory receptor to increase the membrane permeability to sodium ion, and this sodium ion will cause large influx of sodium influx resulting in depolarization of the membrane, and then you can see the membrane potential has risen to minus 45 millivolts. The part C, you can see here it is an inhibitory type of neuron causing the opening of the chloride channel, and this chloride influx will result in decrease in the resting membrane potential to a lesser way, to more negative form, that is minus 70 millivolts. So when we talk about EPSP here, it has caused the shift of a electric potential from minus 65 to minus 45. That is, EPSP is plus 20 millivolt. And when we talk about IPSP, it has causes the membrane potential from minus 65 millivolts to minus 70 millivolt. Therefore, IPSP here will be minus 5 millivolt. So let us understand with the help of this animation the difference between EPSP and IPSP. This is a normal neuron and the potential here being minus 60 millivolt. Excitatory neuron causing influx of sodium and the potential is depolarization in nature, EPSP. And when inhibitory neuron will be there, influx of chloride, the potential will be hyperpolarizing type, it is IPSP. So you have seen EPSP and IPSP. So the common features between the two are, first, they are graded potential. They are not action potential, but they need to be summated, reach a threshold value, and then there will be firing of action potential. Second, they are decremental in nature. They can be over a period of time. For example, here you can see the inhibitory neurons are acting, but after a distance from the dendrite to the soma, they have decremented or decreased. Similarly, for the excitatory, the electrotonic potential, they are localized, and after traveling for certain distance, they then tend to disappear. Now talking about EPSP or the excitatory postsynaptic potential, the neurotransmitters are glutamate or aspartate. So as we said, it will cause excitation in the neuron which is on which it is synapsing. So which channel do you think will cause to that depolarization? It will be the sodium ion channel. Whenever there is excitation by the neuron on the postsynaptic membrane, it will result in excitation of this channel. Here you can see neurotransmitter is binding to this sodium ion channel, resulting in opening of the sodium ion channel, influx of sodium ion. This increased concentration of sodium inside the neuron will result in decrease in the negativity or the increase in the positivity inside the cell, resulting in the depolarization of the postsynaptic membrane, which is termed as EPSP. The EPSP so formed, resulting due to the opening of the sodium channel, result in generation of EPSP, that is just plus 8 millivolt. It helps to facilitate the transmission of the nerve signal. As we said earlier, it is subthreshold. It is not an action potential. It can be summated. It is localized, and it is decremental, meaning that first it will have a latency, reaches a particular amplitude. Here it is 8 millivolt, and after some time it will decay. That is, after 15 milliseconds it tends to decay. So if we want to summate one potential with the another potential, the time lag should not be more than 15 milliseconds. And similarly, when we talk about the inhibitory postsynaptic potential, the neurotransmitters which are inhibitory in nature are glycine and GABA. They will act on this channel to cause increase in chloride influx, as we have talked earlier, increase in efflux of potassium. Potassium is more inside the cell. So whenever there is opening of this potassium channel, it will result more in movement of potassium from the inside to the outside, leading to more negativity inside, which is known as hyperpolarized state. These are due to either the opening of the chloride channel resulting in influx, or opening of the potassium channel leading to the influx, and both these conditions will cause what? They will cause the generation of IPSP, which are again they can be summated, non-propagated, subthreshold in nature, decremental in nature, having a magnitude that is -2 millivolt, and they will decay after some time. So basically, the soma is acting as an integrator. It is receiving the excitatory neurotransmission, EPSP. It is receiving the inhibitory form, that is IPSP. So soma is ultimately acting as an integrator so that it can summate the responses so received from a neuron. But a single discharge from a single presynaptic membrane can never cause an action potential to reach to the value of plus 20 millivolt that is responsible for the firing of action potential, meaning thereby that many of the synapses are to reach to the synapse and soma, and then they will be firing. For example, here you can see this diagram. If only 4 synapses are firing, it is subthreshold in nature. When we have 8, you can see the potential is EPSP, but when 16 neurons and synapses are firing, it leads to the generation of positive potential, maybe plus 20 millivolt, that results in the firing of the action potential. So this synaptic integration is the essence of the synaptic transmission. So summation occurs in the soma itself. So the net postsynaptic potential will be the EPSP minus the IPSP, and if it is in positive direction and if it is in sufficient amount, there will be generation of the action potential and transmission of the nerve impulse. So here you can see the resting membrane potential minus 60, the excitatory are coming, causing the change in the membrane potential, the EPSP, then again another excitatory causing EPSP, then again another excitatory E3 causing again excitation of the neuron. So finally all these will then summate if they fire together or fire in an interval before the decay of the neuron. So what will happen? It will cause the generation of action potential. A single neuron will not result in the generation of this potential. Here you can see the threshold being there, and the single firing does not result in generation of action potential, but two have fired simultaneously, and then we get an action potential which can then be transmitted along the neuronal length. And if we have all three firing simultaneously, again a threshold will be reached, so it will fire. But if we have inhibitory and the excitatory, again it will be sub-threshold only, because EPSP minus IPSP, the net postsynaptic potential which will be generated, will decide the fate of a nerve impulse. So summation can be a temporal summation or the spatial summation. Just by looking at this figure, what do you see? That's temporal summation. There is only one postsynaptic neuron, a presynaptic neuron, and firing of the presynaptic neuron after a repeated period result in summation. Whereas in spatial summation, we have different numbers of the presynaptic neuron which are converging onto the one postsynaptic neuron, resulting in firing of action potential. So temporal is nothing but repeated stimuli are applied over a short period of time. First stimulus adds to the next, to the next, to the next, so it results in summation of the action potential. Whereas spatial, we have number of neurons which converge on a single postsynaptic neuron, resulting in membrane potential. So synaptic integration, here you can see A single neuron, B a single neuron, excitatory in nature but still subthreshold, C inhibitory in nature, A plus B both have added together resulting in firing of action potential, A plus C excitatory and inhibitory bringing the membrane potential back to normal. And the part of the membrane which is responsible for excitation of this membrane potential or generation of postsynaptic potential is initial segment. Why? Because this has greater number of the sodium channels and it is unmyelinated nerve fiber. The number of sodium channels are highest here. So whenever a potential of 60 to 10 millivolt is there, even that results in the generation of action potential. So this action potential, once generated or the inspiratory spike is formed, it forms an action potential. It travels in both the direction, but retrograde transportation, it is faded and only propagates in anterograde direction.